Hello everyone. Today we are going to talk about the Watson and Crick's model of DNA, or to be precise, is the B form of DNA. As you can see over here, that Watson, the two scientists, Watson and Crick, they are working on the model of the DNA, and they are credited with the discovery of the double helical structure of DNA. So they worked in 1953 and came up with the model of the structure of the double helix of DNA. So before we move into the structure of the DNA, let us see where is the location of the DNA. So you can see over here there is a cell, and in the cell there is a nucleus, and inside the nucleus there are chromosomes. And when you open the arms of a chromosome, there is the one where you get the DNA. So as you can see, there is a gene, then there is a nucleotide, then these are the double helical structure of DNA. So let us move ahead. Now, of today, the DNA double helix model of DNA is probably the most iconic of all biological molecules. Now, you can see here towards the right hand side, there are three DNA molecules. You can see they are the double helix. However, on the left, there is just an analogy where a bridge was made, which is pretty much similar to that of DNA molecule. So it shows how the DNA molecule is so important, not only in terms of heredity, but also it provides so much of structural integrity as well. So James Watson and Francis Crick in 1953 combined the physical and chemical data and proposed a double helical model for DNA. And this model is the most widely accepted model for the structure of DNA, and it consists of two strands which are connected together by hydrogen bonds and are helically twisted. So there you have seen all the DNA molecules, irrespective of different forms, they are double helix or helically twisted. Now, if you look at the diagram, there is a 2-deoxyribose sugar is there. As you know that DNA is stands for deoxyribonucleic acid. So here you can see 2-deoxyribose, and if you look at the position of the age group that is highlighted or marked with a rectangular red colored box is the age group that makes it different, and in deoxyribose there is age group. However, when you see compare with the ribose sugar, that in that particular position there is OH group. But please keep that in mind, in deoxyribose there is H group at the 2' carbon. Now, besides this, there are purine and pyrimidine. Those are nitrogenous bases. So purines are adenine and guanine, and pyrimidines are cytosine and thymine. Now, it is often seen that students, they get confused which one is a cytosine, which one is belongs to purines, and which one belongs to pyrimidines. So it's just a analogy. Don't it's not that I'm trying to put it up in a very complex way, rather trying to make it simple. You can just remember by the spelling of pyrimidines. Pyrimidines, basically py is there. So if you see cytosine, cy is there, and in thymine also y is there. So you can remember in that way. The pyrimidine bases are cytosine and thymine, whereas purine bases are adenine and guanine. Okay, so the sequence of bonding is such that for every ATGC, it's a random combination that I've given over here is ATGC. It can be AG, CT and all. So it varies. On one strand, there would be TACG on the other hand. That shows that the two chains are complementary to each other. Okay, so let us see the actual structure or double helical structure of the DNA molecule. If you see in the diagram towards the left, what you see is that the distance between two helix or the diameter between the helix is 2 nanometer, or in some books you'll find that they write 20 Armstrong also. So this is equivalent to your 2 nanometer. Besides this, what you see in this structure is that there are purine and pyrimidine bases, and always remember that A pairs with T. So A stands for adenine and T stands for thymine. So in between A and T, the hydrogen bond, there are 2 hydrogen bonds. Whereas in case of cytosine and guanine, there are 3 hydrogen bonds. And always keep that in mind, that G pairs always with C and A pairs with T always. Now, besides this, if you see the distance between two bases, there is 0.34 nanometer, and in a complete turn of a DNA molecule, there are 10 bases. So you multiply 10 into 0.34, that becomes 3.4 nanometers. So a complete turn of the DNA molecule has 3.4 nanometer distance. Besides this, if you see in the diagram, there are minor grooves as well as major grooves. Now what you see is that there is a sugar phosphate backbone. So if you shift your focus from the left diagram to the right one, what you see is that there is T is there. So T stands for your thymine. So that is a nitrogenous base. The nitrogenous base is connected to a sugar molecule, and sugar molecule is again connected to a phosphate molecule, that is your PO4 3 minus, and then again sugar molecule, then again phosphate molecule. So that's why we are referring to it to as sugar phosphate backbone. So besides this, the two strands are anti-parallel, wherein if you take a closer look, you'll see that one strand runs from 5 prime to 3 prime direction, and the other strand runs from 3 prime to 5 prime direction, and it maintains a polarity. And basically the polarity is basically due to the formation of phosphodiester bond. If you see in the figure towards your right, I have encircled with a blue color, that is PO4 3 minus, that is your phosphodiester bond. So basically phosphodiester bond is a linkage between 3 prime 3 prime carbon of the sugar molecule, of one sugar molecule rather, and it is connected to another 5 prime carbon molecule of the of another sugar. So basically it's a linkage between 3 prime carbon atom of one sugar molecule and 5 prime carbon atom of the another sugar molecule. So this was all about your structure of DNA. Basically it's a bDNA. So I hope you liked it. Thank you very much and have a nice time.